Hi biologists, this is Miss R and we're going to do lab 3.17, feedback in biological systems. The first thing you want to do is pause the video and look around for the link to this MIT Open on feedback in biological systems. It's a very interesting 10-minute video and you'll need to know what's in that video to answer the rest of the questions in the lab. So pause the video and pause this video, go watch the MIT Open video, and then we can continue. Now that you know all about positive and negative feedback loops, we can talk about the differences. One of the differences is that negative feedback loops always are trying to get back to some sort of set point. They use different aspects of the system to always return to this set point here. Whereas a positive feedback system doesn't always come back to this set point, it kind of tends to escalate or amplify the response. Temperature, the way we have homeostasis for temperature in humans is a negative feedback loop. In other words, we're always trying to come back to 98.6. We're always trying to come back to that set point. What are some of the ways that your body maintains a constant internal temperature? How does your body always try to get back to that set point of 98.6 Fahrenheit or 37 degrees Celsius? You can think about it. There are a couple hints in this diagram. What do we do when it's cold out? As Minnesotans, you know the answer to this one. What do you do in the summer when it's really hot? It means you have to drink a lot of water. Those are some good hints. In the video, it talked about how childbirth is a positive feedback loop. And this diagram here obviously is not a childbirth. It's a different positive feedback loop, but it's a good way to illustrate how amplification works. Ethylene gas is produced by, say, one apple on a tree. That signals another apple to produce ethylene gas. Now there's more ethylene gas being produced, which signals more apples to be ripening, and those ripening apples produce even more ethylene gas. So the response keeps going up and up and up, and that's a positive feedback system. In the video, they talked about how childbirth is a positive feedback system. A woman's body produces oxytocin. It makes the uterus contract. And the uterus contracting makes more hormone. And so the response amplifies, of course, until the baby is born. Another example of a negative feedback loop is what you're going to find. We want one besides temperature regulation, which we talked about a few questions ago. Here's a big hint. Your blood sugar is something your body always wants to maintain at a fairly constant level. And we know from the video that diabetics, there's a mistake in that system because insulin is no longer recognized by some of the cells as a signal to pull sugar into the cells and out of the blood. Glucagon is the other side of the system. When you're feeling a little slow and laggy and your blood sugar is maybe a little low because you haven't eaten lunch in a while, your body releases glucagon, which tells your cells to release sugar out into the bloodstream, or your liver more specifically. Now that you know all about positive and negative feedback loops, it's time to think a little bit about population dynamics. There's a second video you need to watch. This one's only a minute and a half, and I'll put that link where you can find it. Now that you've watched the video on carrying capacity, hopefully you understand that this fishbowl is a little bit beyond carrying capacity. No more fish could live in this fishbowl, and that's really what a carrying capacity for an environment is. This fishbowl, on the other hand, you might be able to get a couple more fish in there before it reaches carrying capacity. Here's a good model for carrying capacity. These are things that cause populations to die off: starvation, predators, diseases, human development, loss of habitat. That's for perhaps a deer population here. And of course, annual reproduction is deer being born. And so you can see kind of these are the outs and this is the in. And so the population tends to stay at a kind of a level. It'll vary a little bit, but as the can demonstrates, there's a level which the population usually varies around. So this graph is from the video, and you can see that the population is kind of vacillating back and forth across a constant here. Does this represent a positive feedback loop or a negative feedback loop? And I'll let you think about that for a moment. Now that you know all about feedback loops, positive and negative, and you know about carrying capacity, we're going to talk about how these two factors play out in a lion population. I'm going to give you this URL and you can follow along, or you can just watch what I'm doing on the screen. So if you click on the URL I gave you and click the orange button that says Launch Activity, you'll end up on this screen here. And we're talking about African lions and modeling populations, and we're going to incorporate what we know about carrying capacity and positive and negative feedback cycles in biological systems. So the Ngorongoro crater in Tanzania, Africa, is home to many species of animals, including lions, zebras, wildebeest, and rhinoceros. As scientists, we can describe these groups of animals as populations. We've talked about populations earlier when we talked about evolution. A population is a group of organisms that belongs to the same species and lives in the same general area. Ecology is the study of interactions of populations with their environment. So which one of the following is an example of ecology? Studying earthquakes, looking for planets in outer space, creating pharmaceutical drugs, or studying how global warming affects whales? Make sure you answered the last question before you move on. You can see we're on screen 2 down here. In 1962, unusually heavy rains during the dry season caused a massive buildup of stable flies in the Ngorongoro crater. These flies drained blood and caused skin sores, which infected the poor lions and made them sick. By the following year, the population of lions had crashed between 75 to 100 to just 12. So it went from between 75 and 100 to just 12. And this activity will explore 2 different population models to discover the fate of the lion population. To answer the questions below, we'll use real data from Dr. Craig Packer's research on lions in the Ngorongoro crater. Which of the following questions interests you the most? And there's no correct answer to this question. You can choose whichever question you would like, and then click Next in the lower right-hand corner to continue. Populations change over time. Populations increase when more organisms enter a population, that's immigration, or when organisms leave it, emigration, or when more organisms are born than die. The opposite is true. Populations decrease when more organisms leave than enter, or more organisms die than are born. The data in this graph is hypothetical from 1950 to 1962. Researchers knew that there were about 75 to 100 lions in the crater, but they did not start collecting data until the population crashed here in about 1963. From 1960 to 1963, the population of lions in the crater decreased. Which of the following is most likely to have been true from 1960 to 1963? Well, let's look at the graph. Here's 1960, 1961, 62, 63. So we're looking at the orange part. Do you think more lions are immigrating, coming in, than emigrating, that's going out? Are more lions died every year than were born? No lions have died, or both A and B. Think about it. The graph is going down. What's happening in the population? Now we're going to look at a different part of the graph. Populations change, and we're going to look at this part of the graph here. This is the same hypothetical data from the previous page, the lion population from 1950 to 1963. However, since the lions are isolated in the Ngorongoro crater, let's assume that birth and death rates have a greater influence on the population than emigration or immigration. In other words, the crater is kind of closed off by high cliff walls, so it's hard for other lions to get in or out from other populations. So we're going to look more at birth and death rates. So what is happening here when the population is increasing? Are more lions being born? Are more lions dying? Think about it and choose your answer. What happened to the lions after 1963? After the crater lion population crashed to 12 individuals in 1963, what do you think happened? What do you predict will happen to the lion population after 1963? Explain below. Draw your prediction on the graph to the right starting in 1964. You may either click from point to point. Remember, your prediction starts in 1964. We'll later compare it to real data. So I'm going to let you choose an answer for what you think is going to happen, but I'll give you a hint what's going on in the graph here. Probably, since the stable flies are gone, we're going to see an increase in the population, unless maybe there's a drought or different disease. And I'm guessing the population might kind of return to what it was before. Now we're going to go from lions to bacteria for a moment to think about how populations grow over time. We're going to use a growth model to figure out what happened to the lions after 1963. However, we need to learn about growth models first. We'll use the growth model of bacteria as a simple model. Bacteria reproduce constantly. Let's say we put a single bacterium in a petri dish with some agar food. That's the gel-like material here in this dish here. These bacteria reproduce by cell division once per hour. Each time a cell divides, it gives rise to 2 cells. So what would happen over 12 hours? Well, if we started with 1 right here, after an hour we'd have 2, which barely registers when the graph goes up to 1,000. And then we'd have 4, and then we'd have 8, and then we'd have 16, then we have 32, 64, 128, about 300, so we're kind of starting to register on that scale, about 600, about 1,200, about 2,400, and then about 4,800. So you can see, if you remember from math, this is an exponential curve. It starts to grow very quickly. Wow, we did a pretty good job. Here is the actual graph compared to our prediction. Our prediction was in red and the actual graph is in blue. As you can see, the population starts off growing very slowly right in here, but then it starts to grow faster and faster right in here. Between 4 and 6 hours, the population went from 16 to 32. Well, between 11 and 12 hours, it went from 2,048 to 4,096. This type of growth is called exponential growth. In other words, the function will have an exponent, or one of those little numbers in the upper right-hand corner next to the variable. This is called exponential, or a J-curve. This type of growth occurs when there's enough food, space, and other resources for every single bacterium. If we let this population of bacteria grow in the petri dish for 24 hours, the graph is going to look like this. So it starts out slow, and then it gets a little bit more, and then it just goes crazy. This is why you can get so sick from bacteria so quickly if they get really happy in your body. In which hour are the bacteria growing the fastest? If you're following along and doing this on your own, You actually have to click on the point to pick that point.